Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Over the last two episodes, you met Dr. David Hudson, who works at the intersection of GPCR signaling and metabolic physiology. And then you also met JB, Dr. Johannes Brohagen, whose chemistry and probe design let us visualize these receptors with clarity. Today, we bring them back together, along with Dr. Majellaro, CSO of Celtarys Research, to ask a simple question with big consequences. What happens when a physiologist, a chemist, and a company dedicated to providing GPCR tools decide those tools should be available, validated, and usable? You'll hear how a PhD email shared frustration with unreliable antibodies and a project becoming a full-fledged company grow into ligands probes, distribution, pipelines, and a collaboration model that actually accelerates GPCR drug discovery. We'll talk about trust and fun as real criteria for scientific partnerships and how tech transfer offices can help or slow sharing and why availability is now part of scientific rigor. And if you want sharper calls around efficacy, allosterism, and receptor theory, Terry's Corner is where Dr. Terry Kinakin turns those concepts into short, practical lessons you can use in your drug discovery programs. Visit terrykinakin.com for more information. And because this episode is part of our collaboration with Celtarys Research, you'll hear how their fluorescent ligands help teams move past assays requiring radioactive probes and get cleaner GPCR data faster. This episode is produced as part of our strategic partnership with Celtarys Research. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. And today's podcast episode is a very special one. One, because it's been done in collaboration with our friends and partners at Celtarys Research. And two, it's because we do have Celtarys Research's CSO and co-founder, Maria Majellaro, with us, and two additional guests who you've met in previous episodes. And today we're talking about the power of collaboration and how to together move the GPCR fields forward together. Welcome, everybody.
Hello.
Hi, thanks for the invite.
Yeah, ditto. Thank you and hello.
Thank you for coming back on the podcast. To those of you listening, this seems like a 4 to 6 week launch of podcast episodes, but for some of us, we just recorded these episodes back to back to back. So it's welcome back, JB, David. It was really nice to see you last week, and Maria, as always a pleasure to have you on. Let's start at the beginning, and I want to use this icebreaker question to start us off with the conversation. If you weren't a scientist, what career path would you have taken? And I'll direct this first to Maria, then JB, and then David.
Okay. Yeah, well, actually, I think I would have done something completely different, like, artistic manager or curator or some museum, theaters, because I really enjoy going to concerts or, when I travel, visiting museums and so on. And also because I really enjoy the international environment and, having to discuss with people about your passions. So in this case, science, in that case, would be art. And I think it's also a way, to have some time to reflect about things. And also, let's say, art helps you to better know yourself. And so in the end, it helps also to better you have better relationships with other people around you. So yeah,
that's an interesting one. Would you be an art director, like theater director, or an actress, or
no, director. No, I'm not good for. I mean, I just enjoy watching but I'm not very good at doing this.
But in some sense, you're doing pretty much similar work in the context of science, where you're traveling, you're talking to people, you're understanding what their research is about and what are their pain points.
Mm-hmm.
Yeah, very good way.
In some way.
Wonderful, JB.
Yeah, for me, it was always clear I wanted to go to university and actually wanted to start studying philosophy, which actually then culminated into also science, if you look at it historically. I had a good mentor in my social year, and he said, do something like you can use your hands more on, it's like the pure philosophy would be to drive for you. So I studied chemistry in the end, where I worked with a lot of flasks and compounds to place from A to B. And otherwise, I think what I would have really enjoyed would be like a drawer or something like that. I also like to draw molecules with pen on paper. So maybe like a technical drawer or so would have caught my interest. Like, and if that would have happened, then Maria could have been the art curator and say like it's actually not worth anything. So
We won't have
we could have crossed paths as well. Yeah.
Thank you, JB. Actually, we did have, and the name of our guest escapes me, but we did have a guest on the podcast who is a GPCR scientist who who was in art and got into science because he got into medical drawing and drawing schematic.
Mm-hmm.
I will share the link with you after our recording. And last but not least, David.
Yeah, so my selection is not quite as cultured as Maria and Johannes. So I'm a big fan of heavy plant machinery, and I was quite obsessed with excavators, or diggers as we call them in the UK, when I was a kid. So I would be driving a large excavator, really, hopefully in a gold mine or somewhere similar like that. And I think the analogy for that is, you know, in research, you spend a lot of time digging sort of around in the data to sort of find the gold. Or if you're me, digging yourself out of trouble with various colleagues. Yeah, that would be me. I'm gonna be perfectly happy doing that today.
I love that. And you also can spend time digging into literature, looking from reference to reference to reference that was misused in so many papers, and 10 papers later, you still didn't find the original citation.
The long path to the first reference.
Exactly. All right, for our audience who may have not listened to any of the individual podcast episodes, why don't we go around the room in opposite direction this time? And if you can go very quickly, introduce yourself as to who you are and what is it that you do today. David.
So I'm David Hodson. I'm the Robert Turner Professor of Diabetic Medicine at the University of Oxford. I focus a lot on pancreatic islet biology and G protein coupled receptor signaling, so really the class B GPCRs, GLP-1 receptor, GIP receptor, and glucagon receptor, and so forth.
Thank you, David. JB?
Yeah, I'm Johannes Bruchhagen, or in short, JB. I'm a group leader at the Leibniz Institute for Molecular Pharmacology in Berlin since 2020. I'm a chemist by training. We are using a bottom-up approach, mostly for imaging in more complex settings. So that means making fluorophores and applying them in the cellular landscape in different tissues, with a particular focus on cell surface proteins and within that GPCRs.
Wonderful. Maria.
Yeah, so I'm Maria Majellaro. I'm the Chief Scientific Officer of Celtarys Research, which is a spin-off company of the University of Santiago de Compostela. We founded the company in 2021, but it was my postdoc project. I did my PhD in medicinal chemistry, and we have our own technology. That is a conjugation platform. We apply to develop new tools, basically biological tools that can be then employed to enable the biological assays, let's say, or in general, if we're a way to answer different questions related to, specifically, we've been working on GPCRs, but we work also with other targets. And so you can answer different questions about the GPCR behaviors or screening. We've been developing also assays for screening of new drugs, and
that is wonderful. Thank you, Maria. I was thinking while you were all introducing to yourselves, I was thinking two chemists and a biologist come together in order to collaborate. What happens? But let's start maybe with David and JB's collaboration. If you can share from each of your perspectives, how did the collaboration begin and what kept it going over a decade or more? JB.
Yeah, I was thrown in front of the bus. I had no choice than to collaborate with David because I was in my PhD, and my PhD advisor sent me an email. I think he met David at a conference saying that I can make some molecules. I was working on food pharmacology, so using small molecular switches to control biological function with light, and had it sent to David. I've never heard about really beta cells or insulin, or back in the days at ATP-gated potassium channels. So I wrote David an email, and then made the molecules, shipped it to London back in the days where David was, and then went over myself on a fellowship from the EASD to test them out. And that was the start of a quite successful and trustful and funny collaboration.
Enduring collaboration.
So David, on your end, what was it like?
So I received this email, and JB was parachuted in, arrived on my doorstep, very innocent, good German student, and then I slowly corrupted him over the past 10 years, we've built this kind of research program together, which is actually glued together with shared funding now as well, where we, I think, really work effectively together to sort of the best of our mutual strengths. So it's highly synergistic, and actually JB thinks slightly differently to me. I'll approach things slightly differently, but we always sort of get to a very nice output in the end. So and we're quite good friends, aren't we now, JB? You came to my wedding.
Yeah.
Wonderful. Thank you for sharing that. And Maria, where does Celtarys Research get involved, and how did you meet each of these fine gentlemen here?
We also received an email.
Not a phone call, not a telegram.
Yeah, I think it was May, in 2021, in the middle of 2021. And David contacted us because he wanted to know if it would be interesting in distributing the sending tools. Because they've been contacted by several researchers who wanted to use these probes. And actually the best way to ensure that everyone could have always access to these products was to distribute them by a company. And so then we scheduled our first meeting, and I mean, I knew that they are great researchers. I read their paper and they do very high quality science. But then when I met them, I also understood the collaborative people they are. And so I understood that it was going to be a very nice and during collaboration. So this is basically, so we started then to collaborate, and I mean it grows then in the time, because we always have this continuous communication. Every time there are researchers interested in the tools, we share the questions with them. They answer very quickly. I have to say it's great because we can give feedback. And so we have now a really nice relationship, and we communicate very well.
Thank you, Maria. That's very interesting. And I think I have a follow-up question for David and GB. But before I go there, Maria, maybe at a high level, can you explain how these type of collaborations, and how does for example, an academic lab get to work with Celtarys Research to distribute their tools? What does it look like? From an outside perspective. The reason I'm saying this because I'm sure that there is other Davids and JBs around the world who might be interested in sharing the tools that they create with the community.
So you mean from an organizational point of view?
Yes.
Okay.
Yes.
Yeah. So basically, we established collaborative agreements. So if they are, for instance, universities, we establish these kind of agreements where we set all the different conditions about the distribution and how we are going to produce the compounds and so on. And then basically we let's say do also our marketing activities related to the products, and we basically share all the information with our customers. And as I said, I mean, as a customer, I enjoy when I ask for information. I'm interested in some product to have a quick feedback, and this is what we always do with the customer. So we have a very good workflow related to when we have a request, answers, and also managing all the process. That is, I mean, there are several customers who say, I really enjoy communicating with you because I know you are reliable in that sense. And so we try, I mean, we try to do to give our contribution to that, considering that already the compounds are great, but just to make people easier to reach, to obtain the compounds. So this is basically how we do it.
Thank you, Maria. You used the magic words, I think, the music to my ear. It's process and developing a process in order to get the feedback, but also put products out there in front of everybody else. David, I'm curious to hear, is this the first time where you collaborate with a distributor like Celtarys Research? And where was that moment in your collaboration when you were developing these tools where you said, well, we want to put this out there, this is really a good tool, and figure out how are you going to go about it?
Yeah, I would first like to highlight what Maria said, that like a chemical tool or a probe is just like as good or as shiny as it is available, right? Like that's the absolute essence for the research community as well. And I think that was also the starting point for us to reach out to Celtarys, like knowing their expertise in GPCR research, because I couldn't make my people to fill out FedEx forms like 3 times a week to have the Luxendin shipped to people who requested it. And we were looking for a company that is actually like within the field and quite dynamic. So this is how it started from our end.
Yeah, agreed. It was, we wanted to get the tools out right because we knew they were important and we knew they filled a gap. And we knew that, part of that gap was because the antibodies were not good. And, there was some quite non-rigorous science that was done with those antibodies. So we thought if we can disseminate these tools, then that will be very helpful for the field. We were getting a lot of requests. And, as a research lab, you employ postdocs, technicians, so forth. And we can't have them spend all their time making compound, shipping compound, fielding all these queries. So that was the first thing. The second thing, don't tell my boss, we were never going to make tons of money off this, but it was actually, to get a nice cooperation going with a company where we also learned as well, because we hadn't really done, many endeavors like this previously. But really for us as researchers, what did we want to do? We wanted to work with researchers who knew the stuff and that were providing tools for researchers. And I think that's, really what attracted us to Celtarys versus a lot of, alternatives. They're very GPCR specialized. We met with Maria to discuss. They immediately knew. They said, okay, we can do this. We can solve this issue for you. And it's a relationship, I think, that's evolved really nicely over the last kind of 5 years or so, I guess now. Maria, right?
Yeah, like 4 years, yeah.
Yeah. And then, the hope is for us is that, Celtarys is going to be mega successful. We continue to work with them, and every now and again they'll sponsor some sandwiches at some of our meetings. But
No, that is wonderful. Thank you so much for sharing that. I think it's important as well to highlight that, Celtarys was born in an academic setting. It's a spin-off where science is very important. Not to say that at other companies science isn't important, but I think when you're a small company, you have the ability to very quickly, as you mentioned, Maria, respond and interact and create those situations where there is not a lot of red tape. Mm-hmm. Before you can have an open conversation and explore the possibility of working together.
Yeah, also because, what we enjoy a lot is also that we are supporting this, because sometimes there are some researchers that are interested in other analogs of Luxendin with our fluorophore. So we ship the information to JB and David, and it can be also the starting point for a new collaboration for them. So I think it's very nice for us also to be part of this network. So to be this bridge. So with the same aim in the end. So
yeah, it sounds very much like you directing the play here and connecting people either with tools or with potential collaborators. I know you and I, Maria, recorded our podcast episode together, and we've been partnering since the beginning of this year. But would you please remind our audience about what is Celtarys' mission and how does it support GPCR researchers?
Yeah, so basically our mission is to provide let's say, superior fluorescent ligands to enable robust and sustainable biomedical research. So we have been developing several probes for fluorescent probes for GPCRs, as I said, because this is the main topic in which the research group from which Celtarys raised was focused on. And then we continue to apply the technology to other fields. And basically, we use our technology as an advantage because we can really design these compounds and modify specific points to obtain the best fit between the tool and the target, and also with the assay, the experimental setup you want to employ. And so basically this is, so we have been developing probes for both, for instance, visualization of the expression of targets in cells, also in tissues, but also to set up a screening assays for a new drug development programs.
Thank you, Maria. And I want to highlight as well that on our Dr. GPCR website, you have a very nice collection of articles about all the applications that you can use, the tools that you are distributing and commercializing towards the community. And it's been a great pleasure reading them regularly.
Yeah,
really.
Because we want to stress also the importance of the linker, let's say, because our word is linker, basically. and not just for networking, as I said before, but also linker between functional elements. And so, because we can build these very complex linkers and then apply them through different experiments. Yeah.
Thank you, Maria. All right. Any funny moments or any unexpected or interesting behind the scenes stories during this partnership or while this partnership was being developed? Let's start with you, Maria. Anything? This email 4 years ago from David.
No, I mean, it was a very, let's say, clear email about the needs and so on. So I can say just that I was curious about every time you read an article and you read the names there, you are always curious to know the people behind the names. So I was curious to better know them and establish this relationship. Basically it was that.
Thank you. JB, maybe anything?
Yeah, I guess the fun moments are usually like when we are on video calls, throwing ideas back and forth, but then like also like in a very embracing manner, right? Like it's a very trustful collaboration since a couple of years, and it's just a pleasure to be part of this and to work together. And I think like this is a team that is vibing like on the same wave, so to say.
Yeah.
David, I see you smiling there.
I don't actually remember sending the initial email. You sure that wasn't you, JB? But I'd sort of get hundreds of emails a day, so my memories just fade in as I approach middle age. But it just works right because again we're not talking to business people, and there's nothing wrong with that. It's great. I mean, it's just Maria's a scientist. She's on the same wavelength that we are. We're all sort of learning our way through this as well as things go forward. They sort of had a few changes. We had a few changes. We went to different institutes, right? We've got new legal teams, new TTOs and things like that. So it's just been kind of fun figuring it out as we've gone forward. It's always amusing me to know.
I love it. I love it. Speaking of collaborations, any differences between, in the case of partnering with industry versus academic labs? Any highlights? And I'm asking these questions because not everyone in academia has the opportunity to work with companies like Celtarys or any commercial entities. So what were some of the differences you've noticed compared to collaborating with academic labs? I could speculate, but I really want to hear what your impression has been. And anyone can jump in.
I'll go first and then hand over to JB. I think the key difference is when you are working with a collaborator, you don't have your kind of tech transfer office and sort of IP teams on the other side. So that's another element of kind of learning that we've done as well, and another sort of parallel strand, because those people are really important, because without them we just don't have the knowledge. It's not taught to you, is it, during your sort of postgrad? So they're important for letting us know about licensing, about sort of protections and all these things, and I think that's a key difference. With collaborators you might put MTAs and things in place, but that's pretty straightforward. So that's one big difference, I think, and actually that's another layer of people that are not here today, but I think really make all this go round as well. So particularly being it, am I pronouncing that right, JD? Probably not. And Vimal, who have done a lot of the heavy lifting on their side as well. JB, any thoughts?
I cannot agree more. I mean, this is exactly what I would have said. Like, it's the whole legal part, right? Like, usually, like when you start a collaboration, it's also like chatting to someone at a conference or also shooting an email, right? And then just like you send stuff, maybe you send like sign a standard MTA or so, but like there was a lot of paperwork behind it. And shout outs to the TTOs. They were a great addition to this team, getting these things forward, and still are involved quite a bit, right?
Mm-hmm. Yeah,
it's a really important process, actually, and I think something that kind of you don't think that you're going to learn as a researcher, but I think has actually been quite satisfying and interesting. It's sometimes frustrating as it can be as well. So I mean that's been a good experience as well.
Yeah, at the beginning it may be, more like time consuming, because you have to set all the agreements and so on. But then once you have solved it, it's quite straightforward, I think.
I think the other key thing is when you send a sample to a collaborator, you don't have to put "not for human use," "do not ingest" on the tubes. So then that's sort of, you just trust that they're not going to use it. So this is a key difference as well.
Don't drink.
Yeah, exactly.
And I agree. I think, and to your point, whenever it's the minute there is a collaboration involved with a company, there is the paperwork, there is the tech transfer team, there is the attorneys, and then all that dry language that us scientists, we read it and we're like, whatever, why would I even think about this possibility of something really negative happening? But this is how the world works, and this is the pieces of documentation that have to go into launching such collaborations. Maria, I have a question for you. What do you wish more academic researchers would know about working with companies, like Celtarys Research?
Well, I think that we know that the collaboration between academy and industry is very relevant, because it's not something new, let's say. But if you want that new discoveries can reach, society and daily lives, in the end they have to do this path. So I think it's very important to stress this. So when you see, as JB and David did, when you see that there can be an application of your tools, and it can be, because it's a starting point. Now the Fluorescents are available, a lot of scientists are using them, and so there are new discoveries that are due to the fact that they can use these kind of tools. So let's say the goal is the same. And so I think it's very important to stress this aspect, that it's important also to, as JB and David said, to identify the company with which you want to collaborate. In some cases, it can be better a small company. In other cases, a larger company. It depends on your needs and the application and the kind of users you want to reach. But I think it's a very important aspect to foster this kind of collaborations. And then there is always paperwork to do, unfortunately. But it's very worth it. And this is an example of that. So
I agree. And I think to take it maybe a path beyond the paperwork, it's also important to, just as I asked both JB and David during their individual podcast episodes about how important, what do you look for in a collaborator? And I think one of you responded trust and fun. And I thought that was a really, really nice response. And I think that goes beyond the paperwork. Working with companies like Celtarys Research, trust and fun becomes very important as well, because at the end of the day, it's humans working with humans with one goal in mind, which in this case is putting these tools into the hands of GPCR scientists.
Yeah, also because we are always the same people. The contact for David and JB, it's always me and Wilson now. So you can really establish, this kind of relationship. And so I think it's also because we know all the history of the collaboration and all the things we can do in the future. And so the direct, communication, I think, helps.
Absolutely. Again, going back to you, Maria, when it comes in general, how do you think about handling priorities between science, timeline, and commercialization?
Yeah, well, for Celtarys, what we did is to establish like a department for the management of the portfolio, because if we want that people, as I said, if you want that people can have access to your products and are interested in the portfolio, you have to give some, quick response. And so you have to have a established workflow for that. And so we have a department dedicated to this, where people that manage all the, because it's not, it's a very rigorous aspect, because they all have to manage all the aliquots. The aliquots have to have, everyone has to have the same amount into the vial. You have to be sure about the quality of what you are shipping, the conditions in which you are shipping the compounds. And so there are several aspects you have to manage, as also the advices that you can give and answers to questions related to the application of the compounds. And on the other hand, we have the department dedicated to the research and development. So basically they take care about custom developments, but also internal projects, so in the frame of grants or our internal pipeline. But yes, I mean, we are a small company, so sometimes we have this higher workload, and so we have to shift some people from one side to the other, even though we try to have always, very scheduled weekly activities. And so it's the moment in which I'm very happy. I mean, I'm very proud of my team because they are always happy to switch and do other activities and support and help just for the same aim. So this is the way in which, and also we sometimes drink a beer together. That always helps. Just to put down the stress and say, okay, guys, we can do this.
So you can discuss any issues with any aliquots, any labels, or any catalog numbers.
I think that these are the moments in which you have the best ideas in science in general. So when you are chatting and it's in informal events, let's say, yeah.
Have you ever met in person, the three of you?
No, unfortunately, not yet.
No, I think it's we need to change that. We absolutely need to change that. It's interesting because you're all based in Europe, so it shouldn't be a difficult thing to do.
We started in 2021. It was still COVID. So it was a bit difficult. Now it can be easier. Yeah, maybe in some conferences we will be able to meet each other.
Wonderful, wonderful. Well, Maria, I think you and I also met around that time, and we've never met in person either.
No.
So at least we have an ocean separating us.
Yeah, it's a bit difficult. Yeah.
All right. This is a question for all of you. What's next for your collaborations? Are you looking at developing new tools together, looking into new biology, new applications? Whoever wants to take it on. I did mute you, both of you, JP and David, to reduce the echo. So please go ahead and mute yourselves.
Yeah, I think all of what you mentioned, Yamina, right? New tools, new targets, but always like with the vision to in like more biology and to also provide for the research community. And this is the plan.
Yeah, I mean, I think what Maria said, and something I didn't really think too much about until now, is that the GPCR we work on are highly translational. So if you do some work, it has the potential to be human relevant. So it has added importance actually that you can get those tools out to people that might want them. So thinking about it now, that's a really strong reason, I think, to sort of work with these small, medium enterprises like Celtarys. The second thing is, we do have other things in the pipeline because, of course, we work on Class B GPCRs. They're reasonably interrelated, and a lot of the kind of backbones and knowledge that we've had over the years is also applicable to others. So we've got some new targets coming online, and we're sort of hoping that we can get those over the line in the next few months, right, Maria? And then, whatever we do going forward, because we have to primarily drive the research, right? When we make these things, it is not because we're making them for Celtarys, it's because they're part of the research that we're doing, and then it just so happens that this might be useful as well. Maybe in the future, if we apply for joint funding, particularly the European funding, then actually we can think differently now. What is it we're going to specifically make for the field? So yeah, lots of possibilities.
I think what's very important is like to keep the collaboration, right? The collaboration is always more than the sum of its parts, right? Like if you would ask a pixel about its contribution to the image on the screen, right? It's an idiotic question, right? But like with industry and academia, that we can do more than we could do ourselves alone.
I completely agree.
I agree as well. So what does success look like in the GPCR research community in the next 5 to 10 years, from your perspective?
From the academic perspective, I guess you know the answer, right? Money, fame, publications, third-party funding.
Nature, cell science, 2 R01s, Nobel Prize. I think the GPCR feels massive, right? That sort of, you define it as GPCR biologists, but actually GPCR work just permeates lots of different arenas. Like, we're working with the respiratory people over at Vanderbilt, who are looking for receptors in the lung. I think really it's a really golden time for GPCRs because not only have they become the biggest drug target, now with the incretins they've become even bigger, and they've solved one of the most pressing issues as well. And I think sort of to rephrase it, how are we going to top what has already been done? And I don't think we necessarily have to. I think that it's about iterative understanding in science, and it's just about improving our knowledge of the systems and the complexity of those systems. And I think as we begin to sort of go away from looking at an individual receptor to interactions between the receptors on the kind of molecular scale, this then brings in, a load more information. And I think as JB talks about pixels, my TV has 4,000 pixels, I think. Is that what a 4K TV has, possibly? But, the more definition we have, the better it is. Like when I'm watching soccer or football, as we properly call it in the UK, I want to see the sweat on the player's head, right, as they're taking that penalty. And, that's the level of detail that we need to get on these GPCRs. Not because it's important that the player's sweating, right? But it gives me some context about whether, that player's in a hot or a cold environment or what pressure they're feeling. And I think, all these things are going to be important for the field. And I think the techniques really do exist in the collaborations as well.
Yeah, what I find so fascinating in the GPCR research community is, like, I mean, it's been around for quite some time, right? And you would think, okay, like canonically, like from the beginning, it was like a very simple concept, right? 7 transmembrane proteins sense stimuli from the outside, translated to an intracellular signal. That was more or less it. Then the G proteins came in, and then the beta arrestins came in, and so on and so on. And like now we're looking at atomic coordinates via cryo-EM, right? It's a completely new fields. We're looking at trafficking. We're looking at like intracellular signaling. We're looking at crosstalks and so on. So like even like the concept is so simple to describe. Like there are like so many aspects that like more and more like seeing the light of day. This is where I think is the community going to, right? To grasp the complexity of this rather straightforward to explain receptor.
Yeah.
Maria, anything to add?
No, I think that also I have to say, I mean, I joined the GPCR community in 2018, basically 17. And I always noticed that also the GPCR community is so active. I mean, there are a lot of, cost actions and the networking, and because I think it's also very important to push forward the research in the field. And so I really feel that the GPCR community is so active in that sense, because they know, because of, as JB said, the complexity of all of this. So just we can mention, I went to a conference in Barcelona, the iGPCR Net in July. And so there were very nice presentations about showing how the same GPCR signalizes differently depending on the tissue. So the fact that we always say, no, this receptor is GS coupled, always is not. And so, but it's important to understand that in order to solve this complexity, the collaboration and network is very important. I think that the GPCR community in that sense makes it very well.
Thank you for that. I have a couple of thoughts regarding, what success looks like. I think for me, there's 2 things. Number one, integrating more computational tools and more machine learning and AI tools in order to process the load of information that we can generate today, because we've come a long way. When I started out, we were measuring cyclic AMP with radioactivity by, running the lysates on columns, and, it was this whole debacle. Now you can do it in a fraction of the time. I used to do 24 samples at a time. That's all pretty much what you can run into a day. So that's one, the ability to really start processing and getting insights from the data that we're getting. Number 2, I think getting a better understanding of what happens below the plasma membrane and how are multiple signals, to David's points, integrated across different GPCRs within the same cell, but also within a tissue. And because at the end of the day, the cell has to integrate all of those signals, and then there needs to be a decision made and an equilibrium established at that moment to say, well, this is the response that I'm going to get to. And to your point, Maria, you were mentioning that, a GS-coupled receptor can be GS-coupled to this extent in one tissue, but then may not be GS-coupled in the other one. And being able to, and having the tools to at least to already capture these differences in the lab, but also the tools to process that information on a higher level to get those insights that otherwise we would not been able to do. I think that those are the 2 things that hopefully will bring in my mind, that how the success looks like in the GPCR for the next 5 years. All right. Yes, go ahead. You guess you were going to say something.
No. I mean, undoubtedly, the artificial intelligence will be always more integrated. But I think from that, I mean, it's very important also to stress that we have to continue keeping, experimental data, and because no, I know you are not saying that, but I always think that this integration is very important, but we have to do it carefully. Let's say so.
I agree. It's a tool. It's a tool. It doesn't replace.
Yeah,
it's a tool.
Yeah.
It's I was going to say I can't find the word. It's a tool.
As you said, I mean, to analyze all this data, to extrapolate information, to find correlations, it's a great tool for that. Yeah.
And then adding
you do not look old enough to have done radioactive cyclic AMP assays in 24 sample format. So
thank you. But it was towards the end, because I think I was doing my master's, and then by the time I did my PhD, we switched to the Brett cyclic AMP EPAC reporter. And kind of an anecdote regarding, tech transfer and MTAs. At the time, I was doing my master's working on chemokine receptor signaling, and one of our collaborators was Michel Bouvier. And I was at the University of Montreal at a research hospital, and I had to go up the hill to Michel's lab. And at the time, the easiest way was to send an email, and somebody would give you an aliquot with the DNA and then a printed out map of your DNA sample. And that was pretty much it, what you needed to start a collaboration and transfer an assay from one lab to another. So, but I actually did purify IP3 as well. I even packed columns after spending some time in Michel's lab with one of his postdocs. So, but thank you for the compliment. I'm glad that I don't look old enough to have done those. All right. If you had a magic wand and you can, manifest a new tool to advance the field, what would that tool be like? What would that be? It might be something you're working on currently, or something you wish you will be working on in the future.
Well, I would say, I think that something great would be to be able to study multiple, for instance, multiple signaling pathways or multiple interaction in the same sample. So because you can apply it, for instance, to also not just cell models, but also, let's think also like patient-derived samples and so on. And, it's not, you don't have kilos of samples to use, and also to be able to see how all these different pathways interact between them and so on. So like this magic multiplex assay, that there are already some of them, but I don't think something, for instance, we are very interested in, we are trying to do this, is to try to develop an assay that allows you, for instance, to study both affinity and functional activity in the same experiment. Because every time you see these tables, comparing binding data, functional data, sometimes you have a very good bind, there's no functional activity, the reverse, or maybe you are monitoring the wrong functional path and so on. So I think that having a, universal global vision of that would be great.
I love that, saving time as well.
Yeah. So.
David, maybe you want to chime in?
Yeah, a lot of my time is spent looking at complex tissue, right, and different interactions between different cells, which actually secrete ligands that act on the neighboring cells. So sort of what would be really neat for me going forwards is a way to catalog GPCRs in cells in a complex tissue and see how that differs as a function of their sort of spatial location. So, a little bit like the rainbow mouse, for example, highlighted all the different neurons in multicolor and multiplex. Something a little bit like that. Not figured out quite how to do it yet, but it would be really nice if we could.
That sounds very cool. All right, JB, I'm assuming from a tool perspective, you want to make as many molecules as possible that are specific to the receptor, that then have the right fluorophore and allow, do
Yeah,
some?
partly. I would maybe argue the opposite. Like, I don't want to make a lot of molecules. I just want to actually make like one molecule with one purpose that fits all, right? That is the dream, right? If you have a small molecule that is binding like a neutral allosteric, not even a modulator, but just like a binder, right? That doesn't block the orthosteric site and doesn't lead to any other conformational changes, but just like sticks, doesn't do anything, is small. Does possibly carries a fluorophore, doesn't bleach, gives you information about orientation, right? Of course, 99.9% quantum yield for the fluorophore and so on. Like we're getting more and more there. We have also programs like looking at this, but I think it's important to note that like we tend to think in a tool development thing that like you're always like tackle one by the other, right? But there's like no ultimate solution. If you would say like ideally, like with all the machine learning and AI stuff, you would think like, okay, it's actually quite easy, right? We have atomic structure, we plug it in, we run some scripts or whatever, and then like it spits out the molecule we have to make, but we're not there yet. And that would have been an absolute game changer in my mind.
Agreed. But as Terry Kinakin would say, GPCRs are allosteric machines. So anything that touches the GPCR will change its conformation. But I like the idea of having a compound that you can label different colors with very good fluorophore that allows you to really follow the receptor with minimally disturbing its function.
Right.
All right, I am mindful of the time, and I want to maybe take us towards the end of our conversation. What advice would each of you give to scientists looking to collaborate across disciplines and sectors? Who wants to take this one?
The advice I would like to give to for a researcher that likes to collaborate. First, like know like in which direction you want to go, right? And then just reach out and talk, right? It usually becomes pretty clear pretty soon, like that you get along, right, personally, scientifically, and that actually like your collaborator can provide something that ensures a win-win or win-win-win situation. Ultimately, I think that's the important point, right? Coming back to like find pixels that make an image and not create noise.
Thank you,
your department chair will sort of have a different view to sort of what you might have, and the department chair view will be focus on your own research program and don't get too distracted because you need to define yourself. I actually look at it the other way, right? The collaborations have completely changed the way that I do science and have led to the research profile and program that I have today. So I would say actually throw caution into the wind, go for it, because science is collaborative, right? And ultimately, enjoy and profit from it. But as JB says, it's all about, matching people up. It's not like dating or something like that, but you've got to have fun and you've got to respect the person that you're working with. For all our sort of, apparent stupidity, we are serious scientists and we do sometimes have serious discussions. And that's really, I think, important, and it's what drives us, to remain in academia as well. So, I'd say just get stuck in would be my best advice. But obviously, if it goes completely wrong, for any of your listeners, do not blame me. Blame your department chair instead.
maybe to add something real quick, like you would think like, okay, collaborations is also like interdisciplinary or international, but like a good collaboration is also like your own research group, right? Like where you meet people, where you discuss things, and where you do form actually like a team that wants to achieve something. So that's probably the best place to start, right? When you come to a new group, find good collaborators in your place already.
Yeah, well, people are very important in that sense. I just would like to add for the international collaboration. As you said, get the sense of the vibes, let's say. So if people are cooperative, easy to work with them. But also something I think it's important is to understand the priority of the project for each partner. Because maybe for you it has a high priority, for other people in collaboration it has not the same priority as for you. And it makes that everything slows down and that in the end it doesn't work. So it has to be also something that has the same importance and priority for each one participating in the collaboration.
That is actually very important as well. I agree with everything that you said. And GP, I really like what you mentioned around, collaborating within your own group, especially if you have an interdisciplinary group, where you have chemists, you have biologists, and then people get to learn to talk to each other, but also work well together. All right, last question of this conversation. If there's one thing you want listeners to remember from our conversation today, what would that be? And this is for all three of you. And maybe I'll start. I'll make it for all four of us while you think about it. What I think people should remember is collaboration is, first of all, human. We are scientists. We want to answer scientific questions, and the best way to do it is together with people we enjoy working with, and who also challenge us, challenge our thinking, and challenge our way of looking at our own science, and bring in their expertise in their discipline to make our science and our collaboration move forward better.
Yeah, I would come back to what I mentioned before, is availability, right? Availability of plasmids, availability of chemicals, of probes, and so on. Like, it's quite tedious for a lab, like if you publish like a study and you're using these compounds to then also provide a lot of other people. Like, I happily do so, but I do not have the capacity to just rerun these pipelines. So if you're something that is worthwhile sharing, like to a lot of potential collaborators, potential customers or whatever, yeah, get in touch with industry, seek ways to give people access.
So my 4-year-old son has just finished nursery or creche or kindergarten or whatever you call it over in the States. And they have a phrase, sharing is caring. And I think actually in terms of science, as JV said, it's hard to do because we've got so many different pulls and drivers, finances, staff development, staff training. So this collaboration allows us to do that, but also it really expands the impact of the work and the impact of the tools, because it gets them into the right hands and the right labs to actually do the work. I'm not going to look at GLP-1 receptor in bone because it's not my system and I don't understand it and I'm not the right person. But now somebody can do that, for example. So it really just adds multiples of impact and layers to the work that you do. And I'm not saying everybody's got to do it. I mean, this formed very organically for us. But if the opportunity arises, and bear in mind, part of it for us was about the capacity issue, just go for it. It's been a really good experience.
Thank you.
Yeah, and I would add that, as I said, the collaboration between industry and academy is crucial, and from several points of view. So let's say for making tools available for all the users, but also for the translation of, discoveries to daily lives and, society. But also another important point that is something that I always say about, what I want for Celtarys is that, try to also, when you buy a reagent or something like that, that's the one that are purchasing you the compounds should be also your collaborators. So let's say we are happy to answer people's questions every time they say, have you ever used this tool in this experiment? Do you have some publications you can share with me? Can you support me? Can you share with me some protocols and so on? Because I think it's very important to give that added value to the collaboration to make, let's say, life easier to people that develop the protocols and so on, because we know that all these experimental activities are so hard, and so all the help you can have and all the information, so request the information, let's say, and feel, as a collaborator who is providing you the reagents or so on.
I agree. Thank you so much. Thank you, everyone. I had a lot of fun talking to each of you separately, but also had a lot of fun talking to the three of you during our conversation. Thank you again. I want you to stay on the call while I stop recording, but I really enjoyed it. Thank you so much for your time today.
Thank you to David, JB, and Maria for taking us inside the chemistry, the biology, and the collaboration that turned a simple email into tools the GPCR community now relies on. And if this conversation sparked ideas, share this episode with a colleague. Make sure you're subscribed on Apple Podcasts, Spotify, and YouTube. Leave us a quick review. It helps more GPCR scientists find the podcast. For deeper grounding in receptor pharmacology, Terry's Corner is where Dr. Terry Kanakian turns concepts into practical tools for your drug discovery projects. And if today's discussion on tool availability resonated, explore our Celtarys Research partner page. Their fluorescent ligands and linker technology are powering cleaner assays and faster decisions across GPCR projects. This is our final episode for 2025. Thank you for listening, learning, and being part of this community all year long. We'll be back with new episodes mid-January 2026. And thank you to the Dr. GPCR production team for making these conversations possible. All the links are in the show notes. And because receptors don't work alone, neither should you. Until next time, stay safe.